All right, everybody. Welcome to the exam 3 review videos. We're going to start with chapter 12, which was cell cycle and cell cycle regulation. I apologize, I'm a little under the weather, so my voice is a little raspy. Anyway, so when we talk about the cell cycle, what we're really talking about is two sort of distinct phases, and that's illustrated sort of down here. We have these two phases called interphase and division phase, or M phase. Sorry, I'm going to be clearing my throat a lot. So first of all, we see interphase and we see division. So we know that these cells are going to be dividing. And first we just got to understand sort of why a cell would divide and why it would have to grow. And basically you need that to live. That's how you grow stronger. That's how you can still live on even though your cells are dying. It's because they're dividing, they're reproducing. So there are two types of division: mitosis with a T and meiosis with an I. And meiosis is really your gametes, which are your sexual cells. So that would be sperm cells and egg cells. And we're going to talk about that next chapter. That's all of chapter 13. But mitosis is what we're sort of going to be talking about mostly in this chapter. And mitosis takes one cell and it creates two identical cells to it. So it splits. It goes from one cell like this, and it becomes two cells. So it cuts it in half, but it also is going to grow, and it's going to have the same genetic material, so it's going to be identical. That's really important. And these are going to be your somatic cells, so your non-sex cells. So down here, we have this little diagram, and we see G1, S, G2, and M. You also see G0 over here, which we'll get to later in the video. But this is the cell cycle. So these first three sort of segments, G1, S, and G2, are going to make up interphase. And interphase is the cell sort of growing, replicating its DNA, and preparing for the M phase up here, which is division, which is cell division, and also cytokinesis, which is the actual cutting of the cytoplasm. So these first three phases here make up interphase. And then the M phase is where mitosis, meiosis, and cytokinesis is going to happen. So we're going to start by talking about interphase because it's super important. So that first phase that I called G1, it's also called GAP1. And basically, this is just the cell growing, doing its normal metabolism. It's going to produce new organelles. It's going to produce new proteins. And then there's going to be a checkpoint at the end of G1 going into the S phase that's going to sort of make sure the cell is going to be able to properly replicate its DNA. So after that checkpoint and after G1, we're going to go into the S phase, which is the synthesis phase, and it's going to be replication of DNA. So before we move on, there is a lot of vocab that sounds very similar regarding DNA. So we're just going to start from the bottom up. So DNA, you may remember, it's the double helices that are made up of nucleotides, and DNA is what codes for traits, and those coding regions are called genes. So when you have this DNA, so you have this double helix, it's going to sort of wrap around these histone proteins, and it's going to sort of condense a little bit and create chromatin with an N at the end, chromatin. So chromatin is more condensed than regular DNA, but it's still packed together. And there are two types of chromatin. There's euchromatin and heterochromatin. The euchromatin is usually going to be the more active DNA. So there's going to be more genes being expressed from euchromatin as opposed to heterochromatin. So then we have this chromatin, and when we take that chromatin, which is just kind of like a loose mess of stuff, and we pack it even tighter, it's going to form something like this, and that's going to be a chromosome. So chromosomes are sort of the physical things that you would see in a karyotype graph. So chromosomes are defined by a structure that has a single centromere, and the centromere of a chromosome is the middle sort of region which allows it to bind to things. So this would be one chromosome. But if I just add on another little chromatid, which we'll get to in a second, this is still just one chromosome, but it's 2 chromatids. So chromatids down here are individual copies of DNA. And when you have 2 identical chromatids, they're called sister chromatids. 2 sister chromatids joined at 1 centromere, like down here, is 1 chromosome because it has 1 central region, but it's still 2 chromatids. So it's really helpful to make sure you have that vocab down and you know these clarifications. Another word, and I'll have a vocab sheet at the end of this chapter to go over every vocab word that I think is necessary. Humans have 23 pairs of homologous chromosomes, which means we have a total of 46 chromosomes, but you have 23 from dad and you have 23 from mom. And the thing is, let's say the 5th chromosome you get from dad. So 5th from dad is going to code for the same genes in the same locations as the 5th chromosome that you got from your mom. So they're identical in the sense that they have the same genes. 5th from mom. But they aren't going to be exactly identical. They just code for the same things. Right. So now that we have gone over that, we're going to resume and go into the S phase. So the S phase is called the synthesis phase, and this is where DNA is going to be replicated. And we'll go over the actual process of DNA replication in chapter 16. But the important thing to know is that the amount of DNA doubles. So you double the amount of chromatids because your chromosomes going into the S phase look like this. There's that 1 centromere, and here's the 1 chromatid. But during the S phase, they're going to end up looking like this, those characteristic X's. And these are identical. These are perfectly identical. But they still only have this 1 centromere, which means it's still just 1 chromosome. So I wrote that out here. The number of chromosomes is staying the same. 1x, or you could also see that 2n. I should, well, we won't. We'll get into n later. Ignore what I just said. So 1 copy of one of these chromatids, they'll eventually separate when we get to cell division and mitosis later in the chapter. But 1 of each of these identical sister chromatids will end up in each cell. And then the centrosomes will that eventually split and create 2 separate chromosomes. So the last part of interphase is going to be GAP2, or the G2 phase. So this is after synthesis. So we have double the amount of chromatids, and GAP2 is really going to just be preparing for mitosis. And if we're preparing to split the cell in half into 2 identical cells, we need to do a couple things. One, we need enough size to be able to make 2 fully functional cells. We also are going to need to double the number of organelles, because let's say we only have, obviously you have many mitochondria, but let's pretend that a cell only has 1 mitochondria. If it wants to split and divide, it's going to have to make a second mitochondria, 1 for each cell. So we're going to double the organelles. And then we are also going to have this G2 checkpoint, which is, we've heard that with the G1 checkpoint. So at the end of G2, we're going to have another checkpoint, and we'll go into that more later on the whiteboard. So after that checkpoint, we are ready to move into mitosis. But real quick, here's just sort of a quick sketch of what the phases of interphase look like. So we have this G1 phase, and we have these sort of this 1x DNA, and it's going to remain 1x DNA, and the number of organelles is sort of, we're synthesizing new organelles, but you'll see this cell has grown quite a bit. And then during the S phase, we're going to double the amount of DNA. So what looked like this at first, when we had all these chromosomes, are now going to look like this. You see, we've sort of replicated each individual chromosome and attached it to each other. So the number of chromosomes are the same, but we just doubled the chromatids. And then G2 is going to just be more growth, so this cell's going to get really, really big. It's going to duplicate organelles. So here's, we have an endoplasmic reticulum here, but we have another one that's been synthesized here. We have 2 Golgi apparatuses, and this is all in preparation for the division. So now we're going to talk about mitosis. So mitosis is the actual process of dividing the cell into 2 identical cells, and this is going to be the M phase. So just to sort of remember the phases, I always think PPMAT. PPMAT stands for prophase, prometaphase, metaphase, anaphase, and telophase. So we're just going to go through each of these individually and talk about them. So the first step in mitosis is prophase. And that is going to move these centrosomes, not centromeres, centrosomes are going to move to opposite sides of the cell. And these are our centrosomes. Now, centrosomes are going to be the things that eventually pull these sister chromatids apart. And so we need one on each side of the cell because we want DNA to get pulled to each side of the cell so that when I split this cell, I have DNA on both sides. So that's why those centrosomes are moving to the cell poles. Next up, the chromatin, and remember the chromatin is the slightly condensed version of DNA, is going to condense even further into the visible chromosomes. And these chromosomes, remember, have already been replicated. So they have 2 sister chromatids per chromosome. Mitotic spindle formation speeds up, and that is these guys here. These are microtubules, and microtubules are going to be responsible for pulling these chromatids apart and moving them to opposite sides of the cell. And then the last thing in prophase is the nuclear envelope, which is the thing that's holding the nucleus and the DNA. That envelope is going to start to break down and free these chromosomes. So that's prophase for mitosis. Next up, we have prometaphase. Prometaphase is the envelope. So remember, we had that sort of dashed circle containing all our chromosomes. It's now totally going to be broken down. It's gone. And then the spindle microtubules, which I talked about, are going to connect to the kinetochores of chromosomes. And the kinetochores of these chromosomes are sort of the area that you can split the centromere at. So you see this chromosome has 2 things touching it. It's going to have a microtubule attaching to this kinetochore on this chromosome, or this chromatid rather, and another microtubule connecting to this chromatid. And that's pretty much it. Prometaphase, super easy. Metaphase is also a pretty simple one. It is just going to line all your chromosomes up right down the middle of the cell at something called the metaphasal plate, which isn't a structure, it's just sort of a line, the metaphasal line, sometimes people call it. And then this is also going to be where the M or the division phase checkpoint is going to occur here. Again, we'll do all the checkpoints and regulation later in the chapter. Then anaphase is when we are actually going to pull those chromatids apart. So you can see that these spindle fibers that attach to these kinetochores have broken these chromosomes apart and are pulling them to opposite sides of the cell. Also super important to note, microtubules are actually going to be shortening when they pull, right? Because they're shortening, they're sort of retracting. The end of the microtubules that is actually going to be breaking down to move those chromosomes apart is the end that is closer, that is attaching to the kinetochore. So what that means is you're going to have at the very end of our microtubule, let's say we have our chromosome, and this microtubule is attaching to this kinetochore. This area of microtubule is going to start breaking down into its subunits, which are called tubulin. And as it breaks down, the next portion is going to just keep on binding and pulling this chromosome. So we're going to end up like this, and then these are going to break down, and you'll see how this is sort of pulling us towards the pole. So that's how that happens, and that's through the depolymerization of microtubules on the end closer to the chromosome, the end that's attaching to the chromosome. So that's anaphase. Now, the last phase of mitosis, but not the last phase of the M phase, confusingly, is telophase. And telophase just means that once these chromosomes have all been pulled to their poles, telophase is going to remake that envelope around each set of DNA. It's going to decondense the chromosomes, and it's going to break down that mitotic spindle that we made. And that's it for telophase. So we are done with mitosis now. This is the end of mitosis, but it's not the end of the M phase. So the M phase is actually going to end with cytokinesis, which is an actin-myosin ring is sort of going to form around the center of this really big cell, so right here, and it's going to start shrinking and sort of pinching the cell in half and creates what's called a cleavage furrow, which is just sort of this indentation in the middle of the cell. Because remember, a cell is a like, for lack, I mean, it's not a perfect sphere, but it's kind of like a sphere. So you can imagine we're putting something here and sort of squeezing it into two cells, and that's cytokinesis. So here is a sort of very large, very confusing to look at first, but sort of breakdown of every stage in the cell cycle for mitosis. I would go ahead and pause the video. This would be a really good thing to maybe like jot down for your note sheet if this is something that confuses you. But what I've done is I've listed out each phase, and then over here are the key events at each phase. So what you need to know is happening. And then I also have put the number of chromosomes, which remember is defined by centromeres, the number of chromatids, and the number of homologous pairs that we have. And remember, homologous pairs are identical chromosomes. So this is a homologous pair. This would also be a homologous pair as long as this chromosome looks like this chromosome. Not to be confused with chromatids. So that is pretty much it for cell cycle. I also have a vocab sheet that I think is definitely worth studying that just has all those words that sort of sound very similar listed out. And just make sure you know the difference between, chromatid and chromosome, and make sure you know the difference between centromere and centrosome, because they're totally unrelated. So just take a gander at this vocab sheet and just make sure that all of these terms make sense. And so that is actually going to be it for this part of the video. We're going to move over to the whiteboard for cell cycle regulation.